Chapter 16, Anatomy 202. Now, Chapter 16 deals with the special senses. So again, we're going to be looking at the special sense organs. Again, we talked about last time the hypothalamus is that region of the brain that regulates homeostasis. It regulates our body temperature, also regulates our blood pressure. And a lot of this information comes from the sense organs where we have receptors. And then the information will be transmitted into the central nervous system, processed, then it will travel along the appropriate tracks up to the appropriate region of the brain where a response will be disseminated out. So the sense organs initiate somatic, which is body cells, visceral reflexes, our organs like respiratory, heart rate from that standpoint. And again, the whole purpose of it is to maintain homeostasis. Anytime homeostasis is going to be offset, it can lead to disease states. All right, this slide just shows some of the learning objectives, receptors, sense organs, the four kinds of information obtained from sensory receptors that we'll talk about, and neuronal encoding. All right, sensory receptors, again, these are specialized structures that are located on the exterior part of our bodies. It deals with touch, pressure, pain, sensation, proprioception from that standpoint. And then the impulse is going to be transmitted by way of a generated potential into the central nervous system, and once there, it's going to be processed by the association of the inner neurons. Now again, we have accessory tissues that can be associated with our epithelial tissues, our coverings, our muscles, as well as our connective tissue. Now, connective tissue is made up of anything such as blood, ligaments, and tendons from that standpoint. Now, transduction, this is when we convert one form of energy into another. Now, remember the first law of thermodynamics, energy cannot be created, nor can it be destroyed. So we're going to be looking at things such as sensory receptors that deal with things such as temperature, pressure, touch, sound from that standpoint. Receptor potential, these are local electrical changes that occur. Now, anytime we're going to deal with the transmission of an impulse, we're going to have to change the membrane permeability from a resting state that's going to be in a negative millivolt to a positive state to generate a action potential. Sensation, this is awareness that what's going on in our surroundings. So again, the information is going to be filtered up from the spinal cord to the brain, and then it's going to be carried back down by way of our descending or our motor tracts. So again, it's processed and filtered so we won't get overload of information that may not be important. So it keeps us aware of our surrounding environment, our body temperature, and our pH, because any of these things that can go too far north or south, it can lead to detrimental states for that particular individual. All right, look at this slide. When we look at modalities, again, we look at vision, hearing, taste. Now, take a moment and reflect back to the cranial nerves and which one deals with this. We're going to be looking at label line code that allows the action potentials to be transmitted. Now, when we talk about stereotypical, if the same response occurs, the same stimulus should be perceived, okay? So again, we have to identify the origin of it, the location of it, where it's going to go, and how it's going to be modified in terms of the signal representative as it leaves out. All right, so if you look at this diagram, location, receptive field, all right, you can see the prick of the finger. You see your first neuron, second, and third, in which the transmission of the impulse. So the impulses can start in terms of two-dimensional touch or two-dimensional discriminative touch. So if you are touching two different places, you should be able to identify the type of touch that you receive as well as where it's located. Like if an ant is crawling on you across the hairs on your arm. All right, sensory projection. Again, the brain identifies the site of the stimulation, the location of it. And then the projection deals with how it's going to travel into the central nervous system by way of the posterior root ganglion cell that forms a synapse at the posterior horn. So we got encoding that takes place. Again, which fibers are going to be sending it, how many fibers are going to be involved, and then how quick or how fast the response is going to be. Now remember, we indicated that myelinated, unmyelinated fibers has an important role. The myelinated ones fire much faster than the unmyelinated fibers. All right, sensory adaptation. Again, if a stimulus is prolonged, firing of the neurons can get slower and slower over a period of time where it can become adjusted to that particular stimuli. So we got proprioception. Remember, we said that's body position in space. Phasic receptors, these are adapted. You get a quick burst of action potential when the stimulus first occurred. Then you get a quick response and then it stops, such as smell, cutaneous pressures touching on the skin. Sensory adaptation can be prolonged over a period of time. So the neurons get slower and slower over a particular time frame. All right, modalities: thermoreceptors, temperature; photoreceptors, light; nociceptors, pain; chemo is chemicals, sodium, potassium, chloride, calcium; and mechanoreceptors deals with pressure. Again, you got exterior receptors, outside; interoreceptors, they detect stimuli internally; and proprioception is body position in space, where we talked about our alpha neurons, and we talked about the Golgi tendon apparatuses. Now the distribution is going to be based on the type of sensation, the type of stimulus, the strength of the stimulus, whether or not it has to go to the higher brain centers where it then would travel through the somatosensory, into the somatosensory cortex by traveling through the thalamus. But we're looking at taste, smell, vision, hearing, and balance. And remember, hearing and balance or equilibrium is going to be modified or regulated by the vestibulocochlear nerve, which is cranial nerve number 8. All right, general receptors, again, general senses. So they can relate to anything that deals with the body surfaces. It can be consistent of one or a few sensory nerve fibers, and it can spread about the different connective tissues that can be involved. So again, from anatomy 1, you remember squamous cells, simple squamous, dermis, epidermis. So free nerve endings, these are for pain and temperature. They're going to be associated with mucous membranes and the skin. Now remember, mucous membranes are those parts of the body that are exposed to the external environment. Your tactile disc receptors for light, touch, texture, in terms of feeling different types of objects. I can feel an apple, orange, a baseball, tennis ball. And then your hair receptors, and if you can look up here at figure 16.2 at the top, the one on the far right, you can see the hair bulb, the hair shaft that protrudes out through the skin. So they can be, again, coiled around the hair follicles, and they monitor movement on our body. You can look at this slide. It shows you encapsulated nerve endings. They are wrapped around by the glial cells or connective tissue, and they can enhance the sensitivity or be selective of the type of response based on the stimuli that it will receive. So you got the lamina corpuscle, tactile, end bulb. You got your bulbous corpuscles, your Golgi tendon apparatus, and those that's associated with the muscle spindle. All right, tactile or your myoclonic corpuscles, they deal with light, touch, texture. They're found in the derma papillae. The crus, end bulbs in the mucous membranes, the lamina or the proscenium corpuscles, these are phasic, rapidly adapting. They can be located in the deep pressures, the stretch, tickle, vibration, periosteum, the covering of the bone. And your ruffinic corpuscles, again, tonic. They deal with heavy touch, pressure, joint movement, and stretching of the skin. Now, we talked about first, second, and third motor neurons. First order, this is when it enters the posterior horn of the spinal cord, coming from the periphery of the body. It can enter from the head, pons, medulla, via your cranial nerves. Touch, pressure, proprioceptions are large. They are fast, myelinated fibers that allows them to conduct at a very quick rate. Heat and cold fibers, they are small and unmyelinated, so they tend to be more longer lasting. The second order neurons, they would decussate. Remember, decussation means they cross over to the opposite side. So they can travel from the medulla or the pons and in the thalamus. Now, third order neurons, they form a synapse there at the thalamus, but they don't stop there. They would continue to transmit on up into the cerebral hemisphere or the cerebellum, depending upon where the impulse is going to travel. All right, pain. Pain, again, is discomfort due to injury, toxic stimulation. Pain tends to help us to alert our body to a part that has been traumatized or damaged, so it helps us to protect and get out of harm's way. Loss in diabetes mellitus, again, diabetic neuropathy means loss of sensation, pain from that standpoint. Nociceptors of pain. All right, you got fast pain, slow pain. Slow pain is kind of like chronic type of pain or a migraine headache. Fast pain is stabbing, acute type of pain. Again, the fast travel on large myelinated fibers by 12 to 30 milliseconds, slow 0.5 to 2 milliseconds, very long lasting, like I said, dural type of chronic pain. All right, somatic, skin, muscle, joints, visceral, again, chemical irritants. Ischemia is a decrease in blood supply to an area that can cause localized tissue trauma or death. Injured tissues, they release a chemical called bradykinin. It can increase the pain stimulus, makes us more aware of the injured site. It stimulates a sequence of events that takes place that will lead to promote healing of that injured area. Histamines and prostaglandins and serotonin, they enhance the nociceptors' response to the stimuli. Here we see two major pathways to the brain. Again, first order neurons travel by way of cranial nerves 5, 7, and 9, and 10, which is the vagus. They terminate in the medulla. Second ones, they start at the medulla where the synapse takes place, and they will continue to travel up along the ascending tracts to the thalamus. There it forms a third order synapse at the thalamus, and it goes to the postcentral gyri of the cerebral hemisphere. All right, again, let's look at the breakdown of our tracts. Pain signals from the neck moving down. So this can carry somatic pain from the spinal thalamic tract, which carries most significantly for pain pathways. The spinal reticular tract, it carries pain to the reticular formation. Again, activates our visceral secretions, emotions, and our behavior as we respond to the pain. Gracilis fasciculus, they carry signals to the thalamus, to our visceral parts of the body. Now remember, with visceral pain, like the heart, you don't feel the heart, but you can have what is called referral pain. When you have like kidney stones or renal pain, you get pain in the lower abdominal area for bladder, or pain in the lower back. All right, this is a diagram of your projection pathway. Again, the finger prick, it goes up the finger into the dorsal root ganglion, goes into the posterior horn. It decussates a crossover in the spinal cord and travels on the anterolateral. It goes up. As you can see, the red arrows. You can see the reticular formation here, where it decussates and it crosses over. It forms a synapse in the purple area. That's your second order neuron. It's going to travel on its way up until it reaches the thalamus. There it forms a synapse with the third order neuron, and it goes on up into the somatosensory cortexes of the body. Again, I mentioned referred pain. This is visceral type of located pain that the organ itself cannot feel pain, but is referred to some of the anatomical sites along the dermatomes of the skin. So heart pain can be felt in the shoulder, the arms, the spinal cord segments, anywhere from the first thoracic to the fifth thoracic vertebrae. All right, this is just a diagram, a figure of referred pain in the shoulder. If you look at the diagram on the left, A, you can see all the areas of the body that's highlighted in color. That indicates if it's kidney, bladder, heart, stomach, pancreatic type of pain. All right, when we look at the modulations of pain, analgesic, these are pain relievers. Your Schedule 3, your NSAIDs, your ibuprofen, your Advils, and from this standpoint. Endogenous, these are internally produced opiates to relieve pain. Again, you got your enkephalins and then your endorphins or your dynorphins. These are your larger analgesics or your neuropeptides. The oligopeptides are a little bit smaller in terms of your enkephalins, but they are secreted by our central nervous system, pituitary, and our digestive structure. So they act as neuromodulators. They try to block the pain receptors in order, if it's not being transmitted, it gives us the release of pressure. Again, spinal gating stops pain signals before it enters the posterior horn of spinal cord. So now the analgesic fibers, they're going to rise into the brain stem and descend by way of the reticulospinal tract to alleviate the pain signal. Nociceptors, when pain is being felt, it releases a substance called substance P at the second order neuron. Here, it travels by way of the spinal thalamic tract to the thalamus, and there it relays to the cerebral cortex, where we get the awareness of pain. So sometimes, in order to alleviate the sensation of pain, we must block the release of substance P from our second order of neurons. Now, the pathway for pain being blocked, again, the signal by way of the hypothalamus to the cerebral cortex is carried by the central gray matter of the midbrain. It allows both the autonomic and the conscious influence on pain. Remember, autonomic, fight or flight, sympathetic, parasympathetic. Midbrain carries the signal to certain nuclei within the medulla oblongata and the reticular formation center. From there, it can descend. You're going to see the release of serotonin. Your analgesic fibers are going to go by way of the reticulospinal tract. There, the fibers are going to terminate in the posterior horn of the spinal cord at all different levels. All right, so again, by way of blocking the release of substance P, that allows us to alleviate the transmission of the pain by way of the nociceptors. All right, if you look at this slide, figure 16.5, it shows you the hypothalamus, it shows you the midbrain, the cerebral cortex. So one, you get the nociceptors. All right, so again, if you look at figure 16.5, you see the nociceptors. Remember now, if they release substance P into the spinal interneurons, they will then have an impact on the second order neuron that is going to be traveling up to the spinal thalamic tract. There, remember now, the third order synapse is going to take place, and it has to go through the thalamus to get to the somatosensory cortex. The input from the hypothalamus to by way of the cerebral cortex can converge on the midbrain or the mesencephalon. From there, the signal is going to go to the medulla oblongata by way of the reticular formation center. From there, it can be inhibitory to the spinal inner neurons. There, we can secrete enkephalins that are our body's internal analgesics. It can block the pain transmission, and there, by way of inhibiting the transmission of the postsynaptic neurons of the second order neuron with the transmission of pain. There, the descending analgesic fibers are going to form a synapse with the first order pain fibers blocking their release of their neurotransmitter chemicals, and it inhibits the presynaptic signal that then will travel, and that will block pain. All right, again, another pathway, again, rubbing or massaging the injured site. This can inhibit the inner neuron from entering to the posterior horn, and then that will be received as mechanoreceptors in the skin and the deeper tissues. So this is how the massage therapy comes in to help alleviate pain. Also, with the dermatomes on our skin, acupuncture, they put pins in the certain regions on the skin that will interact with those nerve fibers that will be transmitted into the deep areas of our spinal cord and into our different visceral organs. All right, this slide just talks about the learning outcomes that we're going to be looking at. The tongue, we have taste, that deals with sweet, sour, bitter, salty. Now, when we look at bitter, that's going to be located on the posterior 1/3 of the tongue. And most poisons are bitter. And that's why anything that tastes bad, we spit it out before we swallow it, and that helps to cut down. So when people are going to try to poison you, they incorporate poisons into cakes and things like that that hides the bitter taste. We have papillae. These are the bumps that we find on top of our tongue. So they help us to sense the texture of our food. Now, we know anytime that something that feels awkward or does not feel right, it can interfere with how we would taste that food or the perception of the taste of that food. All right, so we have what are called lingual papillae, the bumps. You got your falciform bulbs, folate, fungiform, and your circumvallate. Now, the falciform, they have no taste, but they sense texture. The folate, they are weakly developed, but these are taste buds, and they begin to form about the age 3. The fungiform, they are on the tips and the sides of our tongue, and the vallate, this is the V shape. So it contains up to half our taste buds. We have what are called taste hairs. A lot of times when we drink something too hot, it can destroy our taste buds, but they can regenerate themselves. But we have that sensation that it's hard for us to taste what that particular substance is. So again, we can look at groups from 40 to 60 different taste receptors. We got supporting cells, we got our basilar cells, we got our taste hairs, or our epithelial cells that we look at, and when they're stimulated, they release the chemicals of the neurotransmitter substances. All right, taste, we have our basal cells, our stem cells. They can be replaced every 7 to 10 days. The supporting cells, again, they can form a synapse and play a role in sensory. All right, salty, sweet, sour, bitter, and umami. These are our immediate tastes of amino acids in chicken or beef broth. Sour, this deals with citric fruits, acids. Sweet, our carbohydrates, and salty, our metal ions such as sodium and potassium. Again, taste, again, temperature, texture will influence how we perceive food. A mouthfeel is detected by the lingual nerve in the papillae. Hot pepper stimulates our free nerve endings of pain. We got regional difference in taste depending upon what region of the tongue that particular taste is going to be perceived at. We look at the two mechanisms of actions of pain.